In this module, we'll talk about PCR technology. Now, where did this topic come from? We just got done talking about DNA replication. It so happens that this technology is based on principles of DNA replication. And why we are talking about it? We are talking about it because this technology has had a major impact on human health and also human society. So let's look at this. First of all, let's look at the requirements for PCR. PCR is basically a process in which you replicate a specific segment of a larger DNA molecule. So it is similar to DNA replication, and its requirements are also similar to DNA replication. So what are the requirements? Number one, we need the target. We need the original DNA. We need 2 oligonucleotide primers. I'll show you what do I mean by that. We need DNA polymerase. We need that for replicating DNA in the cell also. We need the raw material, dNTPs, the nucleotides, and we need the PCR machine. PCR machine basically is an equipment that can change the temperature of the sample or the reaction mixture. So let me explain it to you how this reaction happens. I'm going to draw this whole process first on the screen, and then we will look at a shorter version of it, and then we'll also look at an animation. So let me draw it on the full screen. So basically, what we have is a large segment of DNA, and we are interested in amplifying or replicating a specific segment. DNA, as you know, is double-stranded. We'll make the double strand. So this is the region of interest or the segment of DNA that we want to amplify. Now, in order to do PCR reaction, we have to have some information about the segment that we are going to amplify. I'll put down the polarities of this DNA too. Here's the 3' hydroxyl. This is the 5' end. This is the 5' end of this DNA molecule. And here's the 3' end of the other strand. So the basic information we need is the sequence of the nucleotides here and the nucleotides here. So basically, we make a small fragment of DNA, an oligonucleotide fragment. It is about 20 base pairs long, and it is complementary to the bases present here. So let's say A, T. I'm actually going to do it. I need more space, so I'll do it here. So the sequence of this DNA is A, T, T, G, C, C. This arbitrary sequence. So when we design the primer, we will make a complementary primer. So the complementary primer will have a sequence like this. It's a DNA primer, which we use in the PCR reaction, not an RNA primer, which is made by a primase in the cell. So here's the primer. Again, I will point out that this is actually, primers are generally 20 nucleotides long, but we are making this smaller primer just for the sake of simplicity. We'll make the same, we'll also make a primer, another primer, which is complementary, which has a complementary sequence to this part. So the polarities, let's also put down the polarities. The polarity would be 5' and 3'. Here it will be 5' and 3'. So the first step in PCR reaction is denaturing, or just same as replicating DNA in the cell. First of all, you have to separate the 2 strands. So in PCR, we separate the 2 strands by heating the DNA in the reaction mixture tube. So now the 2 DNA strands are exposed. Their information is available to the outside molecules and in specific to the primer. This primer will specifically find this region and hybridize with this segment of DNA. It so happens that what is magical about the number 20, that 20 nucleotide long fragments would be unique to a DNA, specific DNA. So at random, they would not be present in more than 1 locations. So DNA primer will hybridize here, and the other DNA primer will hybridize here. Remember our polarities. This is the 3', this is the 5', this is the 3', and this 3' hydroxyl is available for extension. Here we have a 5' end, here we have a 3' end, and this 3' end can be extended. Now this primer annealing is done at a lower temperature. Say this temperature is about 95 degrees Celsius. This temperature is generally about 55 degrees Celsius. Now we will again heat our reaction mixture. We are heating because the enzyme in which we are using in our PCR mixture is more efficient at a higher temperature at about 72 degrees centigrade. When we raise the temperature, this enzyme becomes active and it starts extending this fragment in this direction, and it will extend this fragment in this direction. So this is one step of PCR, and which is repeated several times, and this one step is basically 3 temperatures that we have exposed our DNA to: 95, 55 and 72. This is about the range of the temperatures. So we started with one DNA molecule. We ended up, after one step, we ended up with 2 DNA molecules. So let's look at this in a different way. Here again, it is a PCR reaction. So we have target DNA. We heat the sample. The 2 DNA strands separate. The primers here anneal. The arrow is the 3' end. So we again raise the temperature. The DNA polymerase extends the primer, and primer gets incorporated into the new DNA strand, and from 1 DNA strand we have gone to 2. We repeat this process several times, and we can make billions of copies of DNA. I will show you how that is done too. We will look at the numbers too, but this is basically the PCR reaction. Again, requirements for PCR: target DNA, 2 oligonucleotide primers. I hope that is clear why we need the primers. DNA polymerase, the raw material which will get incorporated into the new DNA molecule, and the PCR machine, which is basically a fancy machine that regulates the temperature for a very short period of time. We will continue our discussion of PCR in the next module.